Hello students, in the previous videos we have read about the development of dicot embryo and we saw that the dicot embryo will develop from the zygote. Zygote is 2n in number and it will divide into basal cell and terminal cell where further divisions will occur and they will give rise to the suspensor cell, haustoria, terminal cell and further divisions which give rise to the complete embryo of the dicot that will be present inside the seed to get the nourishment and for the further development. Now, today's topic is development of monocotyledonous embryo. How this monocot develop? It is almost same as dicot. Okay. Now see what happens in this case. Once again we will draw our zygote. Okay. Now suppose this is zygote and this is diploid in nature. Clear? Now I am drawing micropyle at this side and chaliceal end at this side. In your book it is given opposite. Okay. Micropyle is given upside and chaliceal end is given downside. So don't get confused. In your book it is written as a basal cell as well as the apical cell. And we are using the terminal cell in place of the apical cell and the basal cell. Okay. Now, in case of dicot, it was divided into two. But in case of monocot, there will be three divisions. That is, the zygote will enlarge in size and then it will divide into three transverse divisions. Okay, three transverse. At first, the first division will occur. I am drawing it here in very small size like this. This will be the terminal. This will be the basal. And then the basal cell divides once again. And then it will give rise to the middle cell also. Now this is your terminal cell. This is middle cell. And this is called as basal cell. Okay. Now see what happens in this case. The basal cell does not divide at all. Now after this there will be no division in the basal cell. Basal cell only give rise to the nourishment to this developing pro-embryo and after that it will degenerate as usual. Okay. So leave this or we can write down there is no division. Okay. But this middle cell and terminal cell will divide. This terminal cell will divide and then it will divide to form cotyledon. Okay. In your book it is written as the terminal cell divides into the two layers L and L dash. Okay, don't get confused with this. It will divide into two layers L and L dash and after that it will give rise to the upper cotyledon and lower cotyledon. That is the upper part of the cotyledon and lower part of the cotyledon. Clear? That is or we can say that if it is upper terminal, it is called as the L dash. If it is lower terminal, it is called as the L. Now this will give rise to the upper cotyledon and this will give rise to the lower cotyledon. Upper and lower and both will give rise to the cotyledon. So this is about the terminal cell. Now how the terminal cell will divide? Terminal cell will divide into the transverse as well as the longitudinal divisions. Okay. Right angle to each other. That is the terminal cell. Suppose if I will draw terminal cell here, they will divide transverse and vertical division and after that they will form the quadrant, mass of a quadrant, then further transverse and terminal divisions, longitudinal divisions and then they will form a mass of a ball, mass of a cell and upper cell. They will give rise to the upper cotyledon and lower mass of a cell. They will give rise to the lower cotyledon. So this is about the terminal cell. Okay. Now see what happens to the middle cell. The middle cell will give rise to suspensor. Clear? As well as it will give rise to many things. Now what are those many things? Middle cell will give rise to suspensor. Along with the suspensor, it will give rise to plumule, radicle, okay, that is called as the coleorhiza and colectile, and after that hypocotyle, epicotyle, whatever things are there for the embryo, each and every part will be developed from the middle cell. Now how this middle cell will divide? Now see, I am drawing it here. Now suppose this is our terminal cell. I told you this will be the mass of a cell and this will give rise to the cotyledon. Now suppose this is our middle cell. There is further division in the middle cell. Okay. Further division, further mass of a cell, and each layer of the cell will give rise to the different part. For example, they will give rise to plumule plus radicle plus hypocotyle, epicotyle and everything. Then they will give rise to a small suspensor. Now this is suspensor along with a basal cell. So what we can write? This is the suspensor along with basal cell. Now their main function is to provide nourishment, and then finally they will degenerate. Understood? Now, how this middle layer will divide? Now, in your book, it is given as the middle layer will again divide into the two layers. That is N and O layer. Okay? And N layer will give rise to the root cap, root hairs, and N layer, O layer will further give rise to the parts of the root. So, this will divide into the different layers, and then it will give rise to the plumule, radicle, hypocotyle and everything. Now see how the division will occur. Now this was our terminal cell. After division it will give rise to this one, cotyledon. Now if the division will be maximum, multiple cells will be produced, and we know the size of a seed is very small. So, as we have read in the dicot, that is the development of cotyledon will shift it towards one side. The same thing will happen here also. The cotyledon will develop and then it will shift to one side. Now, how it will shift to the one side? That is, the whole cotyledon will appear, or they will grow at one side like this. Now, this is called as the cotyledon, and there is a division. Mass of a cell, we can say that. Okay. So now we will not call it as a terminal cell because it has become the cotyledon. Understood? Now see the next stage, what will happen here? If I draw, now this is our cotyledon, and after further division, the portions will come out. Okay, the plumule will protrude. The radicle will protrude from the lower side. So, after the parts of a cell, they will protrude out. They will give rise the structure like this. Clear? Now see what things we can see here. What things we can see here? This portion is called as cotyledon. Understood? Now, there is the formation of a papery sheath. You must be knowing this thing. That is, papery sheath is formed in the monocot, and that is called as the coleoptile and coleorhiza. So, near the epicotyle, a papery layer will be formed. Suppose this red one is the papery layer. Okay. Now, this papery layer is called as coleoptile. Now, what is this coleoptile? It is a papery sheath. Okay. Papery sheath of the epicotyle. Clear? Now, see. When we come to the downside, now then we will get here mass of a cell after the multiple division, and then we will get epicotyle. Clear? Then we come to this portion, because this is a junction, so it is called as embryonal axis. Okay. Now, this is embryonal axis. Then we come to this layer. Now, this portion is forming. Understood? Then we come to the next layer, and this is called as your suspensor. Okay. And see, from here an outgrowth will be there, and this outgrowth is called as, again with a papery sheath, called as the coleorhiza. Okay. In place of a radicle, there is the coleorhiza. In place of a plumule, there is a coleoptile. So, we can write down, in place of radicle, there is a papery sheath. And here, in place of plumule, there is a papery sheath called as the coleoptile. Okay. Then suspensor is this portion. This is the suspensor layer, and this will be the hypocotyle. Understood? So, what things we have seen? We saw outer side papery layer, coleoptile, then epicotyle, then the junction between the hypocotyle and epicotyle is called as the embryonal axis. This we have read in the dicot also. Then hypocotyle, and after that this coleorhiza, outward growth, and suspensor as well as the basal cell. Okay. Now, function of this suspensor cell and basal cell is to provide nourishment, and after that they will degenerate. Clear? So, this is the structure of a monocotyledonous embryo. Now, we will see how the structure will look after the degeneration of the basal cell and suspensor cell. Clear? I am erasing this first portion. Okay, I am erasing till here. Now, we will get this as a big cotyledon. This is the upper part of the cotyledon. Clear? L dash according to the book, and L. Now, M, N, O, everything has been divided, and they have given rise to the root cap, root apex, radicle, plumule, coleoptile, coleorhiza. These things we have got. So now see the structure. After the formation, it should be a little bit smaller in size. Understood? Now we got this structure. Okay. Now this consists of a papery layer, and this is called as the coleoptile. Clear? And there is also a papery layer called as coleorhiza. Clear? Now after that we will get one layer hypocotyle, and we will get epicotyle. After that, this is a layer, and this layer is called as the embryonal axis. So this is a typical structure of the monocot embryo. Okay. Now after this, we will see this structure inside a monocot seed. So let me draw the monocot seed, and then you will be able to see the structure. You have already read about the monocot seed in the junior level of classes. Suppose if I draw the monocot seed like this. Okay. Now see, we all know that the monocot seed consists of an outer layer, and that is outer layer is called as, what is the name of the outer layer? Aileron layer. Okay. This aileron layer is made up of protein. After that, I have to draw this little bit this side and smaller in size, so that you should be able to see each and every part. Okay. Now see, this is the aileron layer made up of protein. Now this is the double layer structure called as the epithelium. So let me draw epithelium. Epithelium is found here also. Now this is the starchy layer. Let me write it with red. Okay, now this whole, this is the starchy layer, and the two together is called as the cotyledon. Understood? After this, we will see this embryo. Clear? Now see, this is a small piece of embryo that I am drawing. You will be able to see here. Now what is this? This is our cotyledon, and this, the whole structure, as I told you earlier, the whole figure. No need to draw here. Understood? So this is a seed, monocot seed. And this is the complete development. Okay. Now this monocot seed get nourishment from the, and sorry, I have written cotyledon, it should be endosperm, na? Contain protective layer, and it is called as the endosperm. Now see children, aileron layer plus starchy layer together is called as the endosperm, and function of the endosperm is to provide nourishment to the developing embryo. Okay. Now it is a monocot. Already cotyledon is there to give the nourishment, But the nourishment of the cotyledon has already been used up. Everything has been used up. Okay? But still cotyledon is present and we are telling that it is a cotyledon to give the nourishment. Now, from where it will give the nourishment? Or why we are telling it as a cotyledon if it is not providing any nourishment at this level? Because the main function of the cotyledon is to provide nourishment. But at this level, the cotyledon is not giving any nourishment. Okay, because the embryo is getting nourishment from the endosperm. Okay, and this nutrient has already been used up. But whatever nourishment or nutrient is given out by the endosperm, secreted by the endosperm, it will be absorbed by the cotyledon first, and from the cotyledon it will be given to this embryo. Okay. So here cotyledon is behaving as an intermediate from the transmission of nourishment from endosperm to the embryo, and that's why this cotyledon is important in this case. Okay, so this is all about the development of monocotyledonous embryo. Now we have left 2-3 more points, that is parthenogenesis, parthenocarpy and apomixis. After that, formation of fruit and seed. Now the formation of fruit and seed is very clear. When the embryo has been formed, we all know that there will be the development of the seed, there will be the development of the fruit. Now how the fruits are formed? They will form from the ovary. The ovary wall will give rise to the fruit. Whatever is present outside the ovule, everything is called as the ovary, and that will give rise to the fruit. Now we can see in the previous classes also, children, you have studied that the fruit has a pericarp layer, and this pericarp layer is divided into 3 layers: epicarp, mesocarp and endocarp. The epicarp is outermost layer, okay, that may be thick or thin. Then mesocarp is the fleshy part of the fruit, edible part of the fruit, and then endocarp is there, which is again a papery layer, okay, that is outer covering of the seed. We can say that in which the seed remains. So this is about the epi, meso and endocarp. And the fruit which contain all these parts, they are called as the true fruit. Like in case of mango, we have seen the outermost green part is called as the epicarp, the orange or yellow color fleshy part mesocarp, and inside we can see the endocarp, and after that we will see the seed. So this is called as the true fruit. Okay, when the ovary involves itself for the formation of the complete fruit. But sometimes what happens, along with the ovary, some other part of the flower also helps in the formation of the fruit. For example, thalamus can also help in the formation of the fruit. Tepals, they can also help in the formation of the fruit. Okay, so these type of fruit in which along with the ovary, other parts are also involved in the formation of the fruit, they are called as the false fruit. Okay, for example, apple. Apple is a false fruit. Clear? So this is all about the development of cotyledons and fruits. Clear? In the next we will see the parthenogenesis and parthenocarpy.